Whoa, why is my camera not on? Hey guys, how's everything going?
Hello.
Hi.
How was your break?
It was good.
Did you get a break? More or less? How many of you took, so I separate, first of all, sorry I'm late. My stat students just kind of let me run over and I lost track of time because I lose track of time with all the screens. I separate hard break from soft break. Hard break is when you really do turn everything off and just don't do anything work related versus a soft break, which you're not doing as much as you were before, but you're doing stuff still. Did you guys take a hard break or a soft break over the holiday?
Soft break.
Soft break? What were you doing, Kelsey?
We had practice. I've been on campus since since December 28th. So.
Oh, okay. So that's not even very much of a soft break, is it? What about the rest of you?
Mine was soft. I was also here like Kelsey.
I'm having trouble hearing. Kara, yours was soft?
Yeah, and I was applying for jobs and stuff like that, so it wasn't very soft.
Quinn, soft or hard?
Soft.
Soft. What were you doing?
Applying for jobs and working out for volleyball.
Rose?
I think I had a hard break.
Oh, good, good. I had a hard break for about 2 weeks where I didn't even open my computer. I cleaned out my garage. I built some things for the house and I just said, I'm done with schoolwork for a couple of weeks. Good. Do you feel recharged after a hard break?
No.
No. Okay, good. That's the level of enthusiasm I'm looking for. Elizabeth, how was your break?
I think mine was a hard break too, but there was like flooding in my parents' basement that we had to deal with and stuff like that. So,
what happened in your parents' basement?
like a pipe burst, something happened, and then yeah, we just had to deal with that. So,
yeah, it did get cold. My chickens, I don't know if you guys know, I have backyard chickens now. They got their first real snowfall, and the first snow, they poked their head out of the chicken coop and then poked their heads back in and said, we're not doing that. And now they're pretty much, snow, no big deal. They're just out there all times of the day, regardless of weather conditions. But it's their first winter, so they had a little bit of adjustment there at the beginning. I have a sump pump that failed one spring a couple of springs ago, and I don't have a finished basement, and the basement flooded because the sump pump didn't work and didn't expel the water that was coming in. So that's total pain. Let's see, all of you are seniors, right? All of you are seniors. Okay, cool. Zach White, you guys know Zach? He's taking this course as an independent study, so he won't be with us during lecture time, but he will be with us during lab time. So don't be surprised tomorrow in lab when he shows up. Unfortunately, I know all of you, so I'm not going to go through the who are you, where are you from, and all that stuff. So why are you taking this class? Quinn, you still go by Quinn, right?
Yes. It's like in the little section of like biology courses I needed to still take. So
You've you need an animal course, don't you?
Yeah.
And Kiara?
I also need an animal course.
All right. Kelsey?
Yep, I would like to get my degree in May.
And all that is standing between you and your degree is Dr. Klawinski. Oh no. Rose?
Same.
Same. Elizabeth?
Need an animal course after ecology. I got to finish strong.
Need an animal course. Okay, so none of you are doing this because you're fascinated by amphibians and reptiles.
You got me.
Okay, well, it's not really that big of a surprise. So one of my tasks today is to impress upon you why you should care about this course more than just I need an animal class so I can get the hell out of Jewel. So that's part of my goal today. So let me pull something up here. One of the first secrets to good teaching is seeing where your students are and either resolving to meet them where they are or somehow pull them to where you are. And so I'm going to try and pull you guys a little bit today. It might not work, but I figure I need to give it the old college try. Okay. Thinking to myself, how can I be this bad at managing screens? I've only been off for a month or whatever. Okay, can you guys see the PowerPoint? Okay, cool.
Yeah.
Okay. So this is also found on the Moodle site, which, did you guys all get onto the Moodle site? Okay, so it's on there also. But basically, I'm going to try and convince you why doing what we're doing is worth doing. So the first thing that you might ask yourself is what is a herp? So this is a course called herpetology, and herpetology basically stems from Greek herpeton. Is basically Greek word for crawling thing. So herpetology is a study of things that crawl. But more specifically, herps are traditionally amphibians and reptiles. But this poses certain problems with herpetology as a field of study because amphibians and reptiles are two different phylogenetic clades. So the amphibians are one independent clade here, and the reptiles are one independent clade, here. And you'll see that in this clade, we have turtles, Tuatara, which is an ancient lineage that lives in New Zealand. Lizards and snakes, crocodilians, and birds are all considered reptiles. These are all in this branch of the evolutionary tree of the vertebrates, and then the mammals are down here. Well, we typically study caecilians, which are these long worm-like amphibians, salamanders and frogs as amphibians, and we study turtles, tuataras, lizards and snakes, and crocodilians as reptiles, but we tend to exclude birds, even though birds are in the reptilian lineage. Birds are just modern reptiles with feathers based on their phylogeny. And so we have herps that include some, but not all, of the members of the reptiles. And then we have the reptiles that are basically include birds as well. And so one of the questions might be, you know, at a school like Jewel, where we try and teach things that are informed by phylogeny, why would we exclude birds from the study of herpetology? And the answer really is just tradition. The ecology of birds is different enough because birds are warm-blooded endothermic organisms, that the energetics of birds are different. They fly, which most herps cannot do. A lot of them have parental care, which very few amphibians and reptiles have, although there are plenty of them that do. There are just a lot of things that are different. But the bigger reason is that just historically, we've viewed the cold-blooded vertebrates different than the warm-blooded vertebrates, and that's carried over into our coursework. So you can go to a big school like KU or K-State, and you can study ichthyology. You guys know what ichthyology is? Study of what? Anybody? You guys don't know what ichthyology is? No. A study of fishes. You can study herpetology. Whoops, can't spell. You could take a course in herpetology, the study of amphibians and reptiles. You could study ornithology. What is ornithology? Anybody? Nothing. Birds. And what is the study of mammalogy? Why do I feel like you guys are all on mute? Because you all are on mute. What is the study of mammalogy?
Mammalogy, mammals, I'm guessing.
Mammals, you guess, yes. So if you go to any of the large state institutions that have like a vertebrate museum on their campus, you will likely find these courses among the courses that you can take if you are interested in vertebrate biology but aren't interested in medicine and things like that. And so ornithology is its own course because even though birds are reptiles, we have traditionally viewed them as being different from the rest of the vertebrates for a variety of different reasons. So just to kind of orient you to things, this animal in the upper left-hand corner here is a caecilian. It's a long, they almost look worm-like, although they can be fairly big and thick. A large worm-like creature called a caecilian. They're fossorial. They're hard to find. They're rare in collections. You kind of have to dig for them in many cases, or sometimes you'll just fortuitously find them on the surface, but they rarely come up onto the surface. You have things like frogs and toads. This is Bufo americanus from Florida, and then we have salamanders. This is Tarica granulosa from Oregon. And then among the reptiles, we have turtles and tortoises. This is a desert tortoise from Texas. We have snakes and lizards. We have this Anolis stratulus from Puerto Rico, and Crotalus adamantius, the eastern diamondback rattlesnake from Florida. We have the tuatara, which is in its own order. This is once again a relict ancient species that only occurs in New Zealand. And then we have so essentially tortoises, snakes, and lizards. This is Varanus komodoensis, the Komodo dragon from Komodo. So this represents 3 different groups of reptiles. And then we have crocodiles and alligators, the crocodilians, which are another group of reptiles. And then, of course, we have birds, which are another group of reptiles. They just happen to not be included in most herpetology classes. So one of the reasons for this is that when we look at the diversity of what we normally cover in a herpetology class versus the diversity of birds, we find that there is a lot of diversity within amphibians and reptiles. And then if we lump in all of the bird species, it would then become a class that would just actually be attempting to cover too many different taxa under one umbrella. It would be hard to stuff amphibians, non-avian reptiles, and avian reptiles all into one big class, because even in the squamata, the lizards and snakes, we have more lizards and snakes than we have of all of the birds, and you're not even talking about the 6,400 species of frogs that we have. So that's another reason that we don't lump the avian reptiles into the non-avian reptile group for the purposes of teaching. When I was an undergraduate, I took an ornithology class. Here's my ornithology book. That's how thick it is. The Life of Birds by Welty and Batista. Here is your herpetology textbook. So would you rather take a book that, course that attempts to cover this, or would you rather take a book that attempts to cover this?
It's a convincing
So,
one.
Green book.
Okay, green book, okay. So that's part of it also, is just, you know, what can you actually successfully get done in a semester? So, but there are other reasons to kind of group herps as a group. So part of it is the diversity of the group. There's plenty of diversity there to study, and the diversity, while there's a lot of species diversity, there's a smaller subset of other types of diversity. So if we want to look at reproduction, for example, we have species within this group, which we refer to as herptiles, amphibians and reptiles, non-avian reptiles. We have some groups that are oviparous, they produce young from eggs. And then we have groups like the rattlesnakes and boids and things like this that are viviparous, that give birth to live offspring. And so there's a variety of reproduction that is useful to study. In some cases, the viviparity is due to actual placental relationships where the developing embryo is getting nutrients from the mother per se, rather than just getting nutrients from an egg yolk. So some viviparous species essentially incubate their eggs inside the body, and the total nutrition for the embryo comes from the egg versus receiving nutrients directly from the female. So for example, in most snakes, the form of viviparity is actually used to be called ovoviviparity, where you're incubating eggs with yolks inside the female, and then the young hatch out as live offspring. But then in the caecilians, these worm-like amphibians, these guys here, there are a number of these species that keep their eggs inside of them during development. The offspring hatch inside the female, and then they feed off of the lining of the oviduct before they emerge as developing young caecilians. And so there's a variety of different kinds of reproduction that are represented by this species diverse group of amphibians and reptiles. Lots of different kinds of mating systems. So we have, among the frogs, we have a bunch of frog species that are called explosive breeders. These are frogs that gather together in these gigantic aggregations, and males are scrambling around trying to mate with females. And it's kind of what's referred to in the literature as scramble competition for mates. We have monogamous species, we have polygamous species. We even have species that undergo parthenogenesis, where they don't have sexual reproduction the way we normally think of it. They reproduce by stimulating mitosis in unfertilized eggs. And so these are all female species. where the females give rise to new offspring without the benefit of a male. So there's a wide variety of different mating systems. Sex determining systems vary a lot in the amphibians and reptiles that we'll study. So in the vast majority of cases, there's genetic sex determination, but in a subset of these organisms, there's environmental sex determination. In most cases, in the amphibians and reptiles, it's determined by temperature. So it's oftentimes temperature dependent sex determination. But then there are rare cases of sex changing species that are capable of changing sex during the course of their lifetime, largely determined by what the ratio of males and females are in the population that you find yourself in. So once again, a wide diversity of different ways of going about determining sex. Because most of these organisms don't generate their own heat metabolically, the thermal physiology of these guys becomes important. They are what we call poikilotherms. They get their body heat from the environment. And so if you want to think about things like global warming, climate change, and things like that, this is a good group of organisms because they're constrained somewhat by what the thermal environment is that they find themselves in. And so when we think about the environment of, let's say that we're thinking about a snake on a mountainside living near a pond. There's sunlight influencing the ground and rocks, thin rocks, big thick rocks. And if you look at the temperatures that are available at the surface of the soil, it's quite hot. At the surface of the rock, it's quite hot. But if you go underneath the rock, it gets considerably cooler. And what happens with amphibians and reptiles under these types of conditions is that they will behaviorally thermoregulate. They will move into the sun, out of the sun, crawl under the rock, depending on what their own body temperature is, such that even if you laid a snake, a tranquilized snake out on the ground and just let him heat up naturally, he would very quickly get so hot that he would die. He would go above his critical thermal maximum. What we see in live snakes is that live snakes shuttle back and forth between environments such that even though they don't regulate their body temperature the way we do by creating metabolic activity to maintain our body temperature, they can still maintain their body temperature within a relatively narrow range of preferred temperatures just by where they place themselves in the environment. Now, as environments change, it might become easier or more difficult for them to do this effectively. And this is why, for example, in the Appalachian Mountains, there are some species of salamanders that only occur at the very tops of these mountains. And as the climate warms, their environments are getting warmer and drier. And because they're salamanders, they need cool, moist environments, which is why they occur at the tops of mountains to begin with, is because those environments are cooler and moister. But as climate changes, the temperature is increasing more at the tops of the mountains. Those environments are drying out, such that the amount of habitat that they have available at the tops of those mountains is becoming smaller and smaller as time goes on, until there's going to come a point where those species that are limited to mountaintops in the Appalachians are going to probably go extinct. And those species that were at lower elevations on the mountain are going to move up, and we've actually observed them moving up and displacing the populations that are at the top of the mountains. So climate change is basically driving species off of the tops of these mountains because as the environment warms up, they no longer have environments that are within their preferred temperature range. And this is of great concern in the Appalachian Mountains of the United States. Especially with amphibians, they have a complex development. So take any frog species, for example. They begin as eggs that are typically laid in the water. They go through part of their life as aquatic living tadpoles. Then they transform into little froglets. They emerge out onto land. and then they spend the rest of their time on the land surface. And because of this, they are exposed to a lot of different environments over the course of their lifespan. Water carries everything that water flows through. So when we think about water as a polar molecule, as it flows through soils, whatever is in the soil that has any sort of charge on it, it's going to be picked up by water, pulled out of the soil, and ends up in things like ponds and streams. And so whatever is on the land ends up in the water, and water is where amphibians are reproducing. And so those early stages of an amphibian's life that are tied to water allow them to absorb lots of waterborne contaminants. And so these species that have complex developmental pathways can oftentimes be good indicators of, for example, water quality. You find in the aquatic ecology literature, there are certain types of insects and certain types of amphibians that are characterized by being in ultra clean, high oxygen content bodies of water. And then there are other species that are tolerant of more polluted, more contaminated water. And so these species can become indicators for how good a water quality you have. Some people have viewed amphibians as kind of canaries in the coal mine, so to speak, of environmental damage. When amphibians start disappearing, something is generally going wrong in that particular ecosystem, because they can be early indicators because they're exposed to a wide variety of environments over the course of their life. They have lots of different body forms. So if you're interested in something like functional morphology, how organisms move, how organisms have evolved a variety of forms in order to handle different types of environmental challenges, amphibians and reptiles are good groups for that. So for example, is this a snake up here in the upper left-hand corner? Oh, Kiara is saying yes, it is not. It is actually a legless lizard. So this is a legless lizard. If you could look at it closely, you would see that it has an eyelid, but it's a legless lizard, and its body, so if you looked inside of the legless lizard, you would see that it still has pelvic and pectoral girdles, but it doesn't have any external limbs. And if you looked at where the pectoral girdles are, the pectoral girdle is right around here, and the pelvic girdle is about here, and the rest of this is all tail. And so this is a group of lizards. We have them in the United States. The United States has a thing called the slender glass lizard. We won't see it in lab. It tends to occur in the southeastern United States that have lost their limbs altogether, and they locomote basically like a snake does. We have strong runners like collared lizards. This one is from Oklahoma in the Wichita Mountains, where there's a particular variety of very nicely colored collared lizards. This is a member of the genus Draco, which are the flying lizards of Southeast Asia, where it has expanded its ribs, and there's a flap of skin that goes between the ribs that allows this thing to basically sprout gliding wings out of its abdomen that allow it to glide from tree to tree. We have things like a top predator, a very heavily built, stockily built Komodo dragon, once again from islands in the south, in the Malay Archipelago, Southeast Asia. Top predators, they get to be 9 to 12 feet long as adults, and they take down goats and water buffalo and things like this as prey. We have geckos, which I study, that are capable of crawling up very smooth things like panes of glass because they have adhesive pads on their toes. There's a group at the University of Massachusetts, Boston, that is developing, they call it gecko skin, but basically gloves and booties that you put on your feet that use the same types of toe pad material that is on the bottom of geckos' feet to allow things like special forces to scale walls and things like this. A guy that I know is working on that. You have things like the basilisk lizard from Central America that has strong locomotor abilities, but also that has these very long toes that they spread out over the surface of the water that allow them to locomote across small bodies of water. People oftentimes call these Jesus lizards because they're capable of walking on water. So this is just within lizards, you have this diversity of forms. We're not even talking about gliding frogs and gliding snakes and things like this. They're ecologically important in that, in this case, the Komodo dragon I keep coming back to, this is a top predator in its environment. It can basically eat anything that lives on the islands that it lives on, including other Komodo dragons for that matter. But then you can also have amphibians and reptiles that are important ecologically, not because they're super big, but because they're super abundant. This is a coqui tree frog from Puerto Rico. They reach densities up to 25,000 frogs per hectare. So that's a hectare is 100 meters by 100 meters. So basically think of a football field, make it square, and then put 25,000 frogs in there. And that's what you have in the forests of Puerto Rico. And when you get that many frogs in one place, they eat a lot of insects as a result of that. And so they can have important impacts on insect abundance, those insects that are then feeding on plants. If there aren't as many insects because you have all these frogs, then that can actually be good for the plant community. Same thing with lizards in Puerto Rico. This is Anolis gunlocki from Puerto Rico. This species reaches similar densities, or the anoles in Puerto Rico reach similar densities of the frogs. And so in Puerto Rico, even though there's not a big, huge top predator, there are these tiny predators that are so abundant that they essentially have the same effects as a top predator would have on the things that are lower down on the food pyramid. Strangely enough, I've been involved in research in Hawaii. This little guy in the lower left-hand corner has been introduced into Hawaii. And in Hawaii, there are no predators on it. And so it reaches huge abundances. And they have a very loud call. And one of the things that has happened in Hawaii is that in neighborhoods where the coqui frog has become introduced, people's property values have gone down because there's such a noise pollution issue in Hawaii. Have you guys ever been to Hawaii? Anybody? If you ever go to Hawaii, one of the things that you'll notice about the Hawaiian Islands is that they just are generally quiet. If you go out into a forest in Hawaii, they lack all the insects and the frogs and the night active birds and stuff like that that you see normally on a continent, such that there's strangely just quiet places, and people actually who live in Hawaii like that. And then you get these 25,000 per hectare frogs coming in and making mating calls. This species is a tropical species. It tends to breed year round, which means that the males are calling year round. And because Hawaii gets plenty of rain, because it gets trade winds coming off the ocean, they get enough rain that they can breed year round, just like they do in Puerto Rico. You will see in real estate listings in Hawaii, and in the listing, it will tell you whether or not the plot of land that is being sold is frog free or not. And you can lose thousands and thousands of dollars in property value because of this introduced little species of frog that's like less than 2 inches. So they're ecologically important, and in the case of the coqui frog, it becomes even economically important because in some cases, these guys can become nuisances. So those are all interesting reasons for studying herps, just kind of things that I find interesting about them. But then there's also a lot of practical sorts of reasons. In most cases, they're small. They oftentimes have easily observed development. They exist in many cases in large population sizes, so you can get enough of them to study. They're subject to environmental variation in terms of things like moisture gradients and temperature gradients and things like this. They're found in a variety of different habitats, and they also have a complex chemical ecology. So this is Xenopus. This is one of the model organisms. So if you think about model organisms, you have things like Cinorhabditis elegans, you have Drosophila melanogaster, you have Arabidopsis, a plant, you have Mimulus, which is another model organism as a plant. We don't have a ton of vertebrate model organisms, but one of the model organisms that we do have is Xenopus. These are aquatic frogs. They live their entire lives in water. And one of the things that's nice about them is this is a male copulating with a female. And once they've copulated, the female will actually develop eggs. And these eggs are really large. And because these eggs are really large, it's really easy to track development from a newly fertilized egg all the way up through, for example, the blastula phase into gastrulation, all the way through the tadpole phase until you get to the adult frog. And so Xenopus eggs are nice in that they're large enough to study with something simple like a light microscope. You guys might recall back when you were in evolution and ecology, when we got to the end of the newt study, the snake newt study, they were doing what we call patch clamping in order to measure the membrane change of the membrane of Xenopus eggs. So what they did was they took the genes out of the snake that were responsible for sodium channels. They put those genes into a Xenopus and then allowed that Xenopus embryo to generate snake sodium channels. And those snake sodium channels were the sodium channels that were resistant to tetrodotoxin. And so then they could patch clamp the egg, the developing Xenopus egg, and measure what the influence of those sodium channels was in terms of being resistant to different levels of tetrodotoxin. But the only reason that you can do that is that these eggs are big enough that you can actually manipulate them in that way. So one of the things that you'll find is that there are certain amphibian, in particular amphibian model organisms that are used simply because they're easy to work with. If you guys are interested in regeneration studies, so looking at how you can regrow different parts of your body. Newts, so there's a newt that occurs here in Missouri. We have examples of it in the collection that we'll look at tomorrow in lab. The central newt, Notothalamus viridescens, is one of these species of amphibians that if you chop off its leg, it will over time regrow a fully functional, entirely just like the original leg. And so Notothalamus viridescens has become a model organism for people interested in regeneration biology because it has the capacity to regrow body parts that are lost through damage. Another reason that you might be interested in amphibians and reptiles, also from the standpoint of why humans might be interested, is they oftentimes have complex chemical ecology. So I have 3 different species here. This is Ptericha granulosa, which we studied back in evolution and ecology. It produces tetrodotoxin as an anti-predator mechanism. This is a harlequin frog, which is found in Central America and South America, this genus. They also produce skin toxins. And then this is a poison arrow frog from Central America. All of these species produce toxins that might be of medical importance to humans. In some cases, in the case of the newt and Atelopus, the harlequin frogs, the organisms themselves appear to produce the toxins. And in the case of the poison arrow frogs, the toxins are derived from their diet. And so when you go to a zoo and you see a poison dart frog, those poison dart frogs are not toxic because they don't feed on ants any longer. So most of the poison dart frogs are ant specialists. They feed on ants, and they take the formic acid from the ants, and they modify that to create the toxins that are found in the skin. Aboriginal people have used these toxins as of the harlequin frogs and the poison arrow frogs have used these toxins in hunting for millennia. But people who do what they call bioprospecting, which is basically looking at naturally derived compounds to do things like developing pharmaceuticals and stuff, they study these frogs to see what the substances are in the skin and what applications they might have for medicine. So, for example, people have been studying bee venom. The venom glands of bees are being explored for basically prompting immune responses and things like this in people. When you look at snakes, there are a variety of different venomous snakes. On the left-hand side here, this is a rattlesnake from the United States. This is a sea snake. This one happens to be from Australia, but there are a variety of different sea snakes that are found around the world. They're closely related to the cobras. In rattlesnakes, rattlesnake venom is what we refer to as a hemolytic venom, meaning that it tends to break down tissues, as opposed to elapid and hydrophiid venoms, which are neurotoxic venoms, which tend to block nerve transmission. And so we have different groups of snakes that have different types of venoms. And when we look at venoms, one of the things that you find, so this is a paper from 2017, when you take venom, in this case it's from a tropical pit viper from Southeast Asia, and run it through something like high performance liquid chromatography, what you will find is that there are lots of different peaks. Each of these peaks refers to a different group of compounds that are found in venom. And so one of the things that we've learned about snake venom is that it's not just one compound that makes up venom. It's actually a soup of compounds. And in this particular paper, they went through and they isolated each of these different peaks from this snake venom. And then they did chemical analysis of them and looked at, and now these are in the wrong order. Oh, I should have, for some reason, I don't have fraction 1 through 5. But for each of these fractions, fraction 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. For each of these fractions, they've gone in and they have attempted to identify what is underneath each of those peaks. So they can collect those pieces, they run off the HPLC, and then once they've collected the substances that are in those peaks, they can actually look at what's in there. So you find things like cytotoxins. These are going to be things that are breaking down cells, galactose binding lectin, serine proteases. So it wouldn't be surprising at all in a snake that has a hemolytic toxin that a large proportion of the substances that are in that venom are things like proteases. So you see serine protease, you have different proteases all down here, continue through the list. Some of these fractions have lots of different substances in them. And so as we learn more about snake venom, for example, we recognize that they are actually collections of compounds rather than single compounds. And many of these compounds we don't know anything about, and they might have applications for all sorts of things that we just don't know about, because this kind of detailed study of the proteins that are made by snakes that are found in venom is a relatively new area of biology that continues to be explored. Yeah, so proteases, enzymes that break down DNA. So lots of stuff in these things that people just never had the technology to look at in the past, and now we do. And so people are. So there are tons of reasons why one might want to study herpetology. And these are just some of them. Questions? Okay, Kara, why are you taking herpetology?
I'm taking herpetology to learn more about. I thought the different chemical. How should I say this? So like how you were talking about the different frogs might have a different chemical makeups than other types of amphibians. So I thought looking more into that would be kind of interesting.
Yeah, I'm surprised over the time that I've been teaching at William Joel College at there's been a rise in people writing scripts for TV programs, and also it appears in books nowadays where people are killing people more often with tetrodotoxin than they ever did before when I first started teaching back in 2000. And I think it's just because the awareness of tetrodotoxin, because of studies that have been done on newts looking at tetrodotoxin, have become more widespread and wide known. It's shocking the number of times that I see tetrodotoxin in like a police procedural on TV. It's like, people are not getting tetrodotoxin from Fisher Scientific and killing one another. It just doesn't happen that way. Yet it appears there in the police procedural. Kelsey, what else? What were you thinking as we went through all of that?
Like the medical practicalities of some of these things, just because that's like ultimately what I'm interested in. Kind of
okay. Yeah, so there are some substances that have actually been derived from some snake venoms that are used to dissolve blood clots, for example, because that's one of the things that certain snake venoms are particularly good at. Unfortunately, on that front, antivenoms are not, and I've stopped recording for some reason, and I'm not sure why. But anyway, we're practically done anyway. What was I saying? One of the unfortunate things about all of this is that antivenoms are no longer being produced by pharmaceutical companies. So if you go, for example, out to Arizona, there's a guy I know there, his name is Dave Duvall. He studies snake envenomation because evidently in Arizona, hold on a minute, I'm having some difficulties with Zoom. Why? Anyway, it's the place in the United States, Arizona is the place in the United States with the most snake bites, venomous snake bites of any place else. And because snake bites in general are relatively uncommon, pharmaceutical companies don't find it profitable to produce antivenin. And so if you get bit by a snake and you go to a hospital, you would want the hospital to have on hand antivenin for the venomous snakes that are found in the area that you live, but chances are you wouldn't find that. You'd be much better off going to a zoo. Actually, I worked at a zoo when I was in grad school, and we had a refrigerator in the herp house. I worked in the herp house, and in the herp house there were a collection of antivenins that would basically allow you to get an antivenin injection had you happen to be bitten by any of the variety of venomous snakes that we held there. And so in the United States, they're uncommon. And in the third world, think about Central America, South America, where there are a lot of venomous snakes, you can't get any antivenin at all. And so it's unfortunate that pharmaceutical companies don't produce those because there just isn't a big enough market. even though there's a real public health need for those things to be in certain places. What about the rest of you?
I thought it was cool how snake isn't a snake.
Yeah. Snake what?
Snake was not a snake. Snake was a limbless lizard.
Snake was a limbless lizard. Yeah. So evolutionarily, so if you're interested in evolution, which of course I am, that's one of the things that I'm interested in with these organisms is evolution has lots of ways of kind of cracking the same nut in a sense. One of the ways that I like to think about organisms is how form relates to function. And we oftentimes see convergent evolution occurring. So we see in all the snakes basically have no limbs. But when you look at the boas and pythons, one of the things that you'll find is that they have vestigial girdles. And in the snakes, they have a vestigial pelvic girdle. Let me go and find, what time is it? Okay, we've got time. What screen are you guys seeing? Am I sharing screen? Oh, there it is. There we go. Okay. So if you type into your web browser Python spur, what you will see is down here, this is the base of the tail. So this is the body. Can you guys see a picture of a snake butt? So what you have here, these are the belly scutes, and the tail begins right here. So this is the tail of the snake, and this is a type of python. And one of the things that you see is you see these little spurs sticking out right here where these arrows are. These spurs are little scales that are covering the surface of a little bone that projects. And the little bone that projects is a bone that is part of a vestigial pelvic girdle that is found in these primitive snakes. And so here's a picture of the actual skeleton. And unfortunately, it's on the small side. But what you can see is that there is attached to the ribs here, there is a little vestigial pelvic girdle that is a part of the snake. So snakes evolved ultimately from legged squamates, the lizards, and then they just went off on this evolutionary path of legless snakes. But then there are examples within lizards where there are occasional groups of lizards that have evolved leglessness within the lizard clade, independent of the evolution of leglessness in the snakes. And the reason for that is that they live in areas where it's much more efficient to locomote without limbs than to locomote with limbs. And so you oftentimes find this leglessness in what we call fossorial forms, forms that are burrowing or live in sandy environments and things like this. And so, yeah, convergent evolution is pretty common across amphibians and reptiles. Thinking back to the whole toxin thing in amphibians, all of those groups of frogs that I was showing you before, these are all in different families. So this is in a different family, this is in a different family, and obviously salamanders and newts are in a totally different group of amphibians. These are all amphibians that have developed toxicity independent of one another. And so once again, from an evolutionary standpoint, one could ask, well, why did these organisms develop these particular types of toxins? And the fact that you find toxin being generated differently in this species as opposed to this species is just a function of the fact that they're separate evolutionary events of toxicity developing. Elizabeth, what did you find to grab onto from all of this?
It honestly kind of reminds me of like Steve Irwin because I used to watch him a lot as a kid, like his TV show.
Uh-huh.
Yeah, and so like I don't know, it seemed like he talked a lot about anti-venom because he had to have it on hand and stuff like that. So I just thought it was interesting that they're not making it anymore.
Yeah, they are not making it anymore. And so can anyone tell me which of these species, can you guys see my PowerPoint? Which of these species is venomous?
I don't know if the Komodo dragon is like venomous per se, but it produces something.
It is venomous, as it turns out. So they're even venomous lizards. I don't have Heloderma up here, but I can get Heloderma. We also have, we'll talk about this when we talk about prey capture. The Mexican beaded lizards and the Gila monster, this is another group of venomous lizards. So when we talk about venoms and toxins, it's not just limited to amphibians and snakes. We also have lizard groups that are venomous as well. Once again, a different kind of venom than the venom that you see in snakes. It's actually derived from a different set of glands. So in all of these snakes, the venom gland is actually a modification of a salivary gland. And depending upon the group of snakes is a different kind of salivary gland that actually produces the venom. In some cases it's parietal gland, in some cases it's a gland in the lower jaw. In the case of the beaded lizards and Gila monster, it's a gland in the lower jaw. And so, yeah, just lots of convergently different ways of arriving at the same solution. All right, other questions, comments? All right, tomorrow, we haven't talked about the syllabus yet. We'll probably talk about that on Friday. So tomorrow in lab, we will meet in lab in person. Bring your masks. The first part of the semester, when it's too cold to go out and do anything in the field, we're going to spend time in lab learning how to identify amphibians and reptiles. So a full half of the class is going to be spent on identification. And so tomorrow I'll have out the amphibians, and your task for a series of weeks is just going to be to learn how to identify the amphibians that are found in Missouri that we have in our collection, so that you would be able to identify them if you see them on a lab bench. And also you should be able to then identify them in the field. After you've mastered amphibians, we'll move on to the reptiles. We'll also do a similar sort of thing where you'll study the reptiles. At the end, you'll be given a test on identification and natural history. And then by the time we're done with that, it will have started warming up enough that the amphibians are starting to breed and things like that. And at that point, we'll be to spend lab time going out in the field, actually finding and interacting with amphibians or reptiles in the field. But clearly, that's pointless at this particular juncture because it's still winter here. So tomorrow we'll meet in White Science 219. Bring your mask, we'll have gloves there for you, and we'll begin the process of getting to know the amphibians of Missouri. Another thing that I'm going to want you to do is to participate in a little research project that I started last year. One area of amphibian and reptile biology that people look at is roadkill. And one of the challenges with roadkill is that amphibians and reptiles will oftentimes be crossing roads, especially during breeding seasons. And as a result of that, they tend to get killed by cars quite a lot. One of the other things that tends to happen is that people like to target turtles, as it turns out. They will literally drive out of their way to smash a turtle in their truck. And so last April, with a reduced automobile activity that was happening as a result of the pandemic, I decided to start going out and doing bi-weekly surveys of roadkill on Highway 210 between 291 and Richmond. And I just drive that 26-mile stretch of road and I record every dead animal that I see. So much fun. And we take them off the road, we remove them from the road and throw them in the ditch so that we don't count them again at some later date. And since we have data from last April, which was kind of during lockdown, as people refer to it, and I've kept it going all through the fall and into the winter, I want to keep doing that throughout the rest of the semester, overlapping the time period that we collected data from last year, to see if, as economic activity picks up, more people are out moving and things like this, whether that has an impact on the number of animals that are road killed. Not all of those animals that we see are amphibians, reptiles. As a matter of fact, now almost all of them are birds and mammals. But during certain parts of the year, the vast majority of those animals that we see road killed are amphibians and reptiles. There was one point last year where there were so many frogs dead on this one stretch of the road that there were literally just too many to count. It was basically just like this kind of greasy slick on the road that was made up of the bodies of literally thousands and thousands of frogs that had been killed as they were moving out of the ponds where they had been hatched. So the frogs went into a pond, they bred, and then the hatchlings went through their development, and then as they were emerging, they crossed the road to get out of the pond and go into this forested area. And literally there were so many of them on the road that it was impossible to count them, and it just kind of made the road surface slick from their crushed bodies. So I want you guys to come out with me and do road cruising. We typically do it on Sunday mornings early while people are at church, because that reduces the issue of safety with, stopping frequently on the side of the road. But we'll set up a schedule for that. And then that's going to be something that we'll write up as your lab report, is looking at an entire year's worth of roadkill data. So part of it will be data that you guys collect, and part of it will be data that I've been collecting over the last however many months it's been since April 5th. I think April 5th was the first time I went out. So anyway, we'll talk more about those things on Friday, but tomorrow I will see you in lab and we will start doing identification. All right, questions? All right, I'll see you tomorrow then.